In this module, we are going to discuss bacterial nutrition. There are a number of bacteria which have a preference in their nutritional requirements. Some of them are very fastidious, whereas others are not so. But why do we need to study about the bacterial nutrition? Well, to begin with, if we know exactly what the bacterial culture requires and we provide it with that, we will have maximum growth in minimum time, and that is exactly what a researcher needs. He requires large quantities of cultures for his studies. Also, if a bacteria grows well on organic carbon compounds and we provide it with inorganic carbon, it is not going to flourish. Hence, the knowledge of the exact nutritional requirements of a bacterium is necessary if we want to grow it in the laboratory and study it, and also for understanding and taking measures for microbial control. Bacteria would require nutrition mainly to build up cell components. So, in order to understand bacterial nutritional requirements, let us quickly revise the bacterial cell structure. The outermost layer in some bacteria is capsule, which is made up of polysaccharides and polypeptides, that is, carbohydrates and proteins. Next is cell wall, which in the case of gram-positive bacteria is made up of peptidoglycan, and in the case of gram-negative bacteria is made up of lipids. We can say that broadly it is made up of carbohydrates, proteins and lipids. Next is plasma membrane, which is made of phospholipids. The next structural components are ribosomes, made up of proteins and rRNA. The bacterial cell also contains pili and flagella, both of which are made up of proteins. Genetic material is present as nucleoid and plasmids, both of which are made up of DNA. Thus, all the bacterial cell components are made up of macromolecules like carbohydrates, proteins, lipids and nucleic acids. The cell mainly requires macronutrients like carbon, nitrogen, oxygen, hydrogen and phosphorus for making these cellular components. These elements are required in large amounts and they account for more than 95% of the dry cell weight. Micronutrients are elements like sulfur, calcium, potassium, magnesium, iron and manganese, which are required in lesser quantities and they make up less than 1% of the dry cell weight. Both macronutrients and micronutrients are absolutely essential for proper cell functioning. The next requirement is trace elements. These include elements like cobalt, zinc, copper, molybdenum and are required in very small quantities. These account for less than 0.1% of cell weight. These elements are normally present as contaminants in high concentrations in water and other ingredients and hence are not required to be added separately to the culture media. The requirement of the trace elements may differ from species to species and depends mainly on the metabolic activities of the cell. These trace elements mainly act as cofactors for a number of enzymes. Many organisms may require certain pre-formed organic molecules which they cannot synthesize. These are called growth factors and are required generally in a very small amount by that particular organism. Some examples of growth factors include vitamins, whose derivatives as coenzymes are non-protein components of many enzymes, amino acids, chiefly for protein synthesis, and nucleotides for DNA and RNA synthesis. These growth factors are mainly required for biosynthetic processes and for the energy needs of the cell. Thus, basic nutritional requirements for a cell are carbon. It is the main component of functional biological molecules like carbohydrates, proteins, and lipids, accounting for 50% of the dried cell mass. Cells differ in their ability to synthesize all of their carbon compounds. Carbon dioxide is an important simple inorganic source of carbon which is readily used by autotrophs. In contrast, heterotrophs use reduced carbon compounds which act as both a carbon as well as energy source. These organisms are extremely diverse in their ability to use the carbon sources. There are some bacteria like the actinomycetes which can use highly complex compounds like paraffin and even rubber as their carbon source. Methanotrophs use organic compounds like methane, methanol, etc. There is hardly any carbon compound which cannot be utilized by some bacteria or other. The next basic nutritional requirement for a cell is hydrogen. Again, a structural molecule and also a participant in process of energy generation. In cells, it is present in the form of water. Generally, all carbon compounds are also a source of hydrogen. It is never a limiting nutrient and it never happens that metabolism is interrupted due to lack of hydrogen. The next basic nutritional requirement is nitrogen. It is mainly present in amino acids, nucleic acids, membranes, cell walls, and most macromolecules. In cells, it is mainly found in its reduced form. It accounts for around 14% of the dry cell weight. It usually is a growth limiting nutrient and an organism cannot synthesize proteins and nucleotides if nitrogen source is depleted. Nitrogen can be obtained from the environment from both organic as well as inorganic compounds like ammonia, nitrates and nitrites. Many bacteria like Rhizobium and Azotobacter can also utilize molecular free nitrogen and fix it by nitrogen fixation. Reduced forms of nitrogen like ammonia and nitrite also serve as an energy source as in the case of nitrifying bacteria, whereas oxidized inorganic nitrogen compounds like nitrites and nitrates serve as terminal electron acceptors in the case of denitrifying bacteria. The next basic nutritional requirement is oxygen. It is again a structural component required in equal amounts in both aerobes and anaerobes. In the case of aerobes, free oxygen is necessary for growth and serves as a terminal electron acceptor. On the other hand, free oxygen is toxic to anaerobes, so they obtain it in a combined form from the substrate. There are 4 forms of oxygen which are toxic. They are singlet oxygen, peroxide ion, superoxide radical and hydroxyl radical. Yet another nutritional requirement is sulfur. It is present in certain amino acids like cysteine and methionine, some B vitamins like biotin and thymine. Intracellularly, sulfur is present in its reduced form. Most bacteria utilize sulfates as a source of sulfur. Some bacteria are incapable of reducing sulfates and have to be supplied with the reduced forms of sulfur like cysteine or hydrogen sulfide. Some compounds also play an important role in energy generation. Some reduced sulfur compounds like hydrogen sulfide, thiosulfate and elemental sulfur are used as electron donors, whereas oxidized forms like sulfates are used as electron acceptors. The next requirement is phosphorus. It is a constituent of high-energy phosphate compounds like ATP, phospholipids in membranes and in nucleic acids. Most bacteria use inorganic phosphates as their phosphorus source, though some can use organic phosphorus compounds like glycerophosphates. Phosphorus can also act as a growth-limiting nutrient. Thus, all microbes mainly require a carbon source, an energy source and an electron source for their proper growth and survival. On the basis of how they obtain these, they are divided into different categories or classes. Microbes can obtain carbon, which is the most important component of cellular macromolecules, either from atmospheric carbon dioxide or from organic carbon compounds in the environment. The former are called autotrophs and the latter heterotrophs. On the basis of how they obtain energy for their life processes, if they utilize light energy, they are classified as phototrophs, and if they obtain the energy from the oxidation of organic or inorganic compounds, they are called chemotrophs. Microbes that obtain their electrons from organic compounds are called organotrophs, while those that obtain electrons from inorganic compounds are called lithotrophs. Often, the above terms are combined to classify the microbes on the basis of their nutrition. As an example, a chemoorganotroph heterotroph obtains its energy from chemicals, its electrons from organic compounds, and its carbon from pre-made organic compounds. Thus, prokaryotes are mainly classified into four major nutritional classes. Let us study one example of each type of organism. An example of photoautotrophic lithotroph would be the cyanobacteria oscillatoria. This organism is photosynthetic and can convert light energy into its chemical energy. Hence, it is a phototroph. It uses carbon dioxide as its source of carbon and hence is autotrophic. It obtains its electrons from water and other inorganic compounds and hence it is lithotrophic. Hence, it has a wide range of habitats including freshwater, seawater and also moist soil. An example of a photoheterotrophic organotroph is the purple bacterium Rhodobacter spheroides. This microbe is also capable of growing photoautotrophically. It is aquatic, often occurring in freshwater lakes in the zones several meters below the surface where light is still present but oxygen is absent. In these anaerobic environments, organic molecules are incompletely degraded to organic acids. Rhodobacter spheroides has adapted its metabolism such that it can take advantage of these carbon sources when they are available. An example of a chemoautotrophic lithotroph is the ammonia oxidizer Nitrosomonas europaea. This microbe is found in many different habitats where ammonia is abundant, including soil, sewage and freshwater. It is especially common in urban settings where high levels of nitrogen compounds are present due to pollution. Nitrosomonas europaea converts ammonia into nitrous acid and in the process consumes oxygen. The energy generated from this process is used to convert carbon dioxide into cellular material. There are a large number of chemoheterotrophic organotrophs and the most common example is Escherichia coli. Very few E. coli, particularly the pathogenic strain, whereas most other strains are inhabitants of intestines of mammals. Here, they use organic carbon compounds consumed by their host as their source of carbon and electrons. Also, they degrade the carbon compounds to generate energy. After we studied the nutritional requirements and nutritional classification of bacteria, let us now move on to discuss the culture media for the growth of bacteria. A culture medium is a liquid or solid preparation which contains all the nutritional requirements of a microorganism and which is used to grow and store microbes. Culture media are employed in the isolation and maintenance of pure cultures of bacteria and are also used for identification of bacteria according to their biochemical and physiological properties. It is often necessary to have a knowledge about the natural habitat of the microbe to design appropriate culture media which fulfill all the nutritional requirements. Culture media can be classified in various ways depending on the consistency, composition and intended use. On the basis of consistency, culture media are classified into three categories. The first is liquid media. This is also referred to as broth and does not contain the solidifying agent agar. In this media, bacteria grow producing a general turbidity and it is not possible to identify the type of bacteria. Liquid media is routinely used for inoculum preparation, dilution techniques and continuous culture techniques. The second type of media is semi-solid media. This media is soft containing 0.2 to 0.5% agar. This type of media is useful in demonstrating bacterial motility. The third type of media is solid media which contains 2% agar and is generally used to study colony morphology, pigmentation and enumeration of bacteria. On the basis of ingredients, culture media are classified as synthetic or chemically defined media and non-synthetic or complex media. Let us first discuss synthetic media. If all the chemical components of a culture medium are known along with their quantities, the medium is called a chemically defined medium or synthetic medium. A chemically defined medium represents the purest and most consistent culture environment. These media are of great value in studying the nutritional requirements of microorganisms or in studying a great variety of their metabolic and physiological activities. They are also useful when it is necessary to duplicate exactly a previous batch of medium or to record the effects of the deletion or addition of a particular substance. If one has to compose a defined medium for a microbe, one should know the exact nutritional requirements of that particular microbe. Let us now discuss the non-synthetic or complex medium. Complex medium is one in which the exact chemical composition of the medium is not known. It is usually prepared from tissue extracts, infusions, peptones, etc. It is particularly useful while studying an unknown microbe whose nutritional requirements are not known and hence a defined medium cannot be constructed. Complex media provides for all the nutritional requirements as it is a very rich medium. The common constituents of complex media are peptones. Peptones are primarily made of peptides and amino acids. They are obtained by digestion of protein-containing tissues of animals and plants such as meat scraps, beef muscle, gelatin, milk protein, and soybean meal. These materials are then dried down to a powder and are then commercially available as a golden granular powder. It is highly hygroscopic and becomes sticky when exposed to air. Since they are crude digests of complex materials, they contain a great variety of organic and inorganic materials, but they may be deficient in certain minerals and vitamins. Peptones serve as source of carbon, nitrogen, and energy. Three examples of brand names of peptones are tryptone, phytone, and simply peptone. A simple peptone solution will support the growth of many organisms. Vegetable peptones are also available that are made from extracts of vegetables which are rich in proteins. For example, soya peptone and pea peptone. The next component is beef extract. Animal tissues like beef muscle, liver, brain, heart, etc. are boiled in water. This causes the soluble parts to go into the aqueous solution. After the removal of excess of fats, it is then concentrated to form a thick paste or dried to form a powder. This powder too is hygroscopic in nature. It is a rich source of carbon and nitrogen and also contains vitamins, amino acids, growth factors, and minerals. The next component is yeast extract. Yeast extract is similar to beef extract except that the source here are bakers or brewers' yeast cells. These cells are allowed to undergo autolysis by heating, which is followed by removal of cell wall. The extract is then concentrated to form a paste or dried to be supplied as powder. Yeast extract can be used in the place of beef extract in culture media. Yet another component is agar. Agar is the major solidifying agent used in bacteriological media. It is obtained from certain marine algae like Gelidium. It is extracted from algae, purified, dried, and supplied as a powder. It is a sulfated polysaccharide composed mainly of galactose and glucuronic acid. It is added in a concentration of 1.5 to 2% to the liquid media, usually after all the components are added to obtain the solid media. Nutrient media is a very commonly used complex media. The composition is peptic digest of animal tissue 5 grams, sodium chloride 5 grams, beef extract 1.5 grams, yeast extract 1.5 grams and distilled water 1 liter or 1000 ml. Nutrient media contains all the elements that most bacteria need for growth and is used for the general cultivation and maintenance of bacteria in laboratory culture collections. In nutrient media, the carbon source is glucose, beef extract is the source of mineral salts, nitrogen, vitamins and peptones in semi-digested protein. Nutrient broth is the liquid medium and is generally used for cultivation of bacteria. When a broth medium is supplemented with agar, it is called nutrient agar medium.